Let's see. So this is the afternoon session on interpretability in biological sciences. We were supposed to have 3 speakers. Debbie Marks from Harvard, unfortunately, is not feeling well, so she canceled at the last minute. We all hope she's feeling much better. She was extremely apologetic and felt guilty that she's not here. So let's wish her well. So that's why we had a little more time at lunch. Anshul Kundaje is going to talk about interpretable models of functional genomics. So he's an assistant professor of genetics and computer science at Stanford. He leads the integrative analysis efforts for their large-scale genomic data generation efforts, things like Encode and the Roadmap Epigenomics project. His lab develops machine learning models of gene regulation, and they've developed a suite of interpretation frameworks, which is going to be interesting for us, to decipher causal sequences of the syntax and regulatory DNA elements. And, you know, basically they're going to train neural nets. And those things are going to learn representations of DNA pathways. And then they're going to figure out how to extract global and local explanations from those neural nets, which is great. And it's what Anshul calls an interpretable lens for genomic discovery, which sounds really fun. Okay, take it away.
Thanks so much, Rich. Can you all hear me properly?
Yes.
Awesome. Okay. So firstly, thanks to the organizers for this great meeting. I wish I could be there in person, but hopefully next time. So like Rich said, the theme of my talk today is on decoding the sequence syntax of gene regulation and functional genetic variation. And we're going to use deep learning models and interpretation methods to try to accomplish these tasks. So just want to start with a brief introduction for those who aren't too familiar with genetics and molecular biology. As you know, there's a ton of diversity in human populations. Some of this diversity is encoded in our genomes. Each of our genomes contain genetic variants. These are locations where the sequences vary across individuals. So for example, this individual has an A at this position, this individual has a C, and so forth. And of course, we carry 2 copies of the genome in each cell in our body. So each of us can have same versions of these letters in the 2 copies, or we can have different versions, right? And so a lot of this variation that we see in genomes often doesn't do anything. It's just sort of random variation, neutral variation. Some of it actually influences completely benign traits, like the color of your hair, height. And there are, of course, variants that can influence susceptibility to diseases and disease progression, like coronary heart disease. So effectively, cataloging genetic variation in human populations is one of the first essential steps to understand the genetic basis of traits and diseases. And over the last several decades, there has been a revolution in sequencing technology. The sequencing machines have gotten smaller. The throughput has grown at a super exponential rate, and this has allowed population scale sequencing. What this means is that we can now basically sequence genomes of millions of individuals. We can do this just to profile genetic variation in healthy individuals, thereby getting a landscape of what natural genetic variation looks like. Here's just some examples. As I mentioned before, each of us has 2 copies of the genome. So this individual has an A and a C at this position. This individual has AA. This individual again has AC. So this is quite nice. We can start cataloging natural genetic variation, but we can also use the same strategy to profile genetic basis of disease. And the way that's typically done is you sequence a whole bunch of healthy individuals, and then you have a collection of cases, individuals that may be suffering from some disease, and you can sequence their genomes, and you can perform simple association studies. These are literally just cataloging frequencies of alleles or versions of these genetic variants in the control individuals in the cases, and you can perform simple univariate association tests to obtain variants that appear to have statistically significant association with any disease of interest. This is a very popular technique called genome-wide association studies. And this has allowed us to, by us, I mean the community, to catalog a lot of genetic variants, some common. By common, I mean variants that are quite frequent in the population. And There are also rare genetic variants present in just less than, let's say, 0.1% of the population. There are even like private variants that only a single individual has, and so forth. So we can catalog genetic frequency of genetic variants. We can catalog their association with different traits and diseases. And this has been a very powerful strategy to really sort of profile genetic landscapes. But then the big question happens, okay, so if I identified a genetic variation that has association with, let's say, coronary artery disease, what can I do with it? So the next question you typically will ask is, if I find this genetic variant of interest, what is its function? Or how does it affect cellular function, or disease function, or human function, right? These are really important questions. And so we're going to focus on this question as to how we can decode genomes and try to understand the potential consequences of individual letters in the DNA, as well as words and syntax and patterns. So to understand the genome, we first have to dive a bit into the functional anatomy of the genome. So here's a very simple cartoon version of this. Here's the cell, here's the nucleus, here's some DNA. We're unspooling it. You can see some of it is wrapped around these proteins, protein complexes. And this is referred to often as chromatin. I'll talk about that briefly throughout the talk. But let's just go into this section of the genome. We've opened it up, and here you see genes. Genes are pieces of the genome that directly code for proteins. They get transcribed and translated to proteins. And what really defines a cell's function is really the repertoire of genes that are expressed. And every cell has its own recipe of genes that are expressed and repressed. Alongside, the expression of genes, if you're precisely controlling which genes get expressed and repressed at which levels in which cells, you need a control system as well. Sorry, just give me 1 second. So the control system that controls gene expression also happens to be encoded in DNA. And they are typically in the form of these control elements or regulatory elements. These are short segments of DNA, and they essentially act as landing pads for various proteins whose job is to regulate gene activity. And what these proteins do is they have evolved DNA binding domains that recognize fuzzy DNA words. And the genomes of these control elements, the sequence of these control elements, have evolved pretty interesting syntax combinations of these DNA words in specific configurations, density, orientation, and they really define the specificity of which proteins can bind in what configurations to at which positions in the genome next to which genes to activate or repress them in different cell types. So a big open question has been, what is the syntax that evolution has encoded in these regulatory control elements? And there are millions of them in the genome. And they're scattered all over the place. The human genome is 3 billion bases. The protein coding pieces of the genome just about occupy about 1.5% of the 3 billion bases. So there's a lot of free space. And these control elements are sparsely distributed all over the genome. Okay, so we're going to try to focus on this question of decoding sequence syntax of regulatory DNA. And the other reason this is important is we're just going back to the original question of disease variation. Genetic variation, as I mentioned before, there are variants that can that are different across individuals, right? So this, one individual has a C, and this is present usually in, let's say, healthy individuals, and the G sequence, sorry, the G letter at the same position, all of a sudden confers some kind of disease risk. So how does that happen? Well, it could happen through many different mechanisms, but you can think of two potential ones that are pretty prominent. One is that the mutation or the genetic variant could disrupt the sequence of the gene itself, which would affect the protein that's produced. Of course, that's going to potentially have pretty devastating consequences. The protein, if it's mangled and messed up, it's going to cause very severe disorders. So a lot of rare diseases are typically caused by mutations inside coding portions of genes that actually tear apart or mutate proteins itself. But a lot of common diseases that typically occur in relatively healthy human populations, mostly affect people in their older age and so forth. They rarely affect gene sequences directly, but they usually affect control elements. So you have mutations present or variants present in control sequences, and they affect the regulation of genes. And you can imagine over time, the system starts breaking down with specific configurations of genetic variants, and in the presence of specific environmental stimuli, this effect can be pretty severe, right? So we're going to try to also understand what these variants might be doing. And for that, we need
Anshul,
data.
can I ask a clarification question? The
Yes.
last slide.
Yes.
If you were to change the spacing of the control elements, does that end up having a large impact?
Yes. It's typically a smooth function. Spacing does have an effect of where the control elements are. It's a pretty flexible distributed syntax in terms of where the control elements lie, but it's basically sort of like a geometric decay function from the start of the gene to where the element lies on average. And so the influence of an element is sort of related to the distance of the element from the gene start site, but not linearly. It's typically more of a power law.
Actually,
Okay.
Is there an evolution reason for this? Like usually the genes seem to be more stable, and then the control elements not as stable. Is that a good way to look at it?
Yes, it actually allows incredible evolutionary plasticity. So, as species have gotten more complex, it's not by creating more and more genes, right? It's a pretty reasonably small repertoire of proteins. 26,000 genes, that's all humans have. The complexity kicks in by really combinatorics, right? So which genes you express in how much, in what levels, in what combinations can give rise to a tremendous repertoire of gene expression profiles. And by increasing or playing around with the complexity of the control elements, you can really change the quantitative levels of genes. And so that provides actually tremendous flexibility. And also by keeping genes protected, right, just through natural selection, you can play around with the control elements in a much more flexible way. So that's one of the reasons why there's such sort of diversity in the control sequences, and there's loose conservation, there's loose pressure to keep them exactly intact, because it allows for evolution and adaptation and so forth.
Compared with Drosophila, like we share 95% of genes and like 70% of the control elements.
That's right. I mean, you can go all the way to yeast, and we share a lot of genes even with yeast, right? So the conservation at the level of genes is very high, but control elements neo-functionalize, and there's lots of unique elements in every species and through adaptation.
Thanks.
All right, so if you want to figure this out, we need some data, right? So I told you where we can get genetic data from, but that doesn't tell us about functional properties of DNA. So luckily what we can do is there's been a revolution in using sequencing technology to actually profile molecular properties of DNA. So for example, this black protein binds these positions to activate these genes. We can perform one experiment that gives you a beautiful profile across the entire genome and tells you where this protein binds. It's like a signal, quantitative readout at every base in the genome. And so you get a readout. And you can do this for all kinds of molecular markers, as I show you here. And these markers can decorate the genome, showing you dynamic activity of the same genome in all kinds of cell types, cell states, healthy, diseased, across individuals, across cell types, and so forth, right? So this has been a very powerful molecular profiling tool. So this is a complement to genome sequencing. And as I mentioned before, when you're interpreting genomes, you need to interpret the genome in the context of different cell types, because all cell types in an individual have the same genome, but they give rise to completely different properties and functions, right? So what NIH has funded over the last almost 2 decades is large projects like ENCODE and Roadmap. What they've done is they've created, just as you have the reference human genome, they've created a reference molecular map of the human genome. And this is not a simple sequence. It's actually a 3-dimensional cube of information, where one axis is the genome. The other axis is hundreds of different kinds of molecular readouts, thousands of proteins, different kinds of chemical modifications to DNA, gene expression levels. And all of this is done in hundreds and thousands of cell types, tissues, individuals, cell states. And now we can do this at single cell resolution. So it's a huge amount of data. And we can use this data to now make sense of how the DNA sequence, this 3 billion base pairs, how does it encode all this diversity of information, right? So we're going to use machine learning models trained on these data to do two things: to decode the sequence syntax of these control elements, and then to use the models as oracles to predict effects of mutations in silico, okay? And also interpret the models and understand what these mutations might be doing. All right, so part one. So our models are very simple. Unlike most applications in machine learning, our neural networks are not super complicated. They're actually pretty simple. Their goal is to take the DNA sequence, 3 billion letters, right? A linear sequence of 3 billion letters with just an alphabet of 4, which is nice and easy, A, C, G, D. So you one hot encode these sequences, and we build convolutional neural networks. They have about 10, 15 layers at max. They're dilated convolutional neural networks with residual connections. They have reasonably large receptive fields. And the goal is to try to map these sequences to profiles of molecular profiles from experiments at single base resolution. So the neural network basically encapsulates a mapping from genomic sequence to molecular activity of a specific readout in a specific cell type, right? So there's one model for every experiment, okay? And once we have this model, we've developed and used several methods for very simple feature attribution. So we have a method called deeplift, which is a fast backprop-based method, similar to saliency maps, but slightly different. We use finite difference propagation instead of infinitesimal gradients. And there are advantages to this approach. We have other approaches, forward propagation approaches like fast ISM. But in the end, what they do is they can take the model, look at a prediction of the model, and try to infer contribution scores of individual bases in the sequence. And so these models, actually, we've applied them to now thousands of data sets, and they happen to be extremely accurate. So here I'm showing you just some predictions from the model. Each of these is 4 different proteins that are binding some location in the genome. These squiggly patterns, think of them as like a shadow of the protein. So you get these little spikes right where the protein binds. And we're trying to predict the shadows of the protein from sequence alone, right? And so I'm showing you predictions here from the model at single base resolution. This is the observed data. This is the predicted data. This is on held out chromosomes. So we train the models on a subset of chromosomes, and we predict on an entirely held out subset. So the models have never seen a subset of the chromosomes, right? And they can do really well. You can see that they can really get these patterns extremely accurately, and different proteins have different shadows, and it can capture in the same cell type. This is in mouse embryonic stem cells. Just looking at 4 proteins, there are thousands of these, and we are modeling all of these, but I'm just showing you 4 of them. You can see all 4 of them bind this element. They all have different shadows. They're binding at slightly different places, right? And if we just evaluate this across the entire chromosome, using Jensen-Shannon divergence metric, comparing the observed profiles to the predicted profiles at single base resolution, then what we see is the red curve is our performance of the model against the ground truth. The blue curve is concordance between replicate experiments. If we did the experiment twice, how similar would it be? That's sort of our upper bound. And then the gray and the green line are various like random, shuffled baseline and an average baseline and so forth. So we are obviously much better than the baseline, and we're approaching a slightly, worse than replicate concordance. So our models are really, really doing a great job pulling sequence features out to be able to predict profiles. Now we can use this, as I said before, we can now start trying to understand what these sequences are doing. So biologists often want to ask, okay, this is a distal control element that regulates this famous gene called OCT4, which is used to reprogram cells. And I want to understand, like, okay, I see this kind of binding signal, right? This activity signal, and these 4 proteins are binding that particular piece of DNA. You can see their shadows right here. We can use a neural network. So there's 4 neural networks or a multitask neural network trained on each of these tasks. And we can just interpret using feature attribution which bases in the sequence are driving binding. And I can use the word driving because there is a causal flow of information from sequence to binding in this particular case, this implicit causality. So you can see that, deeplift, for example, highlights some features that are pretty common to all 4 proteins. And then there are specific features like this one right here, this little DNA word, TG80. It pops up specifically for nanog. And then there are these GGG sequences that pop up specifically for KLF4. And we can map these back to known DNA binding words and really start tearing apart the precise bases in the sequence that are driving binding of different factors, right? So this is very simple feature annotation. Now we can do this for millions of elements in the genome, and we might want to summarize what are the global patterns that the models are learning. What are these discrete sort of DNA words? Obviously, they're being repeatedly used in many, many thousands of sequences. So using feature attribution, we can, of course, interpret individual sequences, but there are millions of them. Can we somehow summarize all the patterns we see, the elemental patterns across all the sequences? So we have a simple distillation framework where we use attribution methods to get contribution scores of every base in every sequence of interest. Then we can effectively throw away pieces of the sequence that appear to be unimportant through the lens of the model. And then we can use a simple alignment and clustering procedure. So for example, we align subsequences that appear to be similar using custom distance metrics. And then we cluster these using graph community detection methods and then we collapse them into consolidated motifs. So this is a very nice way to take the millions of parameters of the net and sort of compress it into a very interpretable set of compact, non-redundant words that are repeatedly being used across these sequences for proteins of interest. And for example, when we do that for these 4 proteins, we pick up a pretty complex repertoire of DNA words required for just 4 proteins to bind. So remember, there are 6,000, right? So it's not that each protein recognizes just one word. There's complex combinatorial cooperative binding. And so to explain binding of 4 proteins, we actually need 50 motifs. And each of them has different combinatorial activity. They have different kinds of patterns. They produce different kinds of combinatorial shadows. And many of them map to previously known motifs. And we also find many novel ones, which I won't focus too much on. There's a very interesting kind of biological stories. We can go into repeat elements and really tear them apart and identify lots and lots of cool biology. And this is published already last year. But I really want to focus on the syntax piece. So far we've been able to annotate individual elements, and we've been able to consolidate into distilled words. But we really want to kind of understand higher order properties. So for example, there's been a lot of debate about the role of spacing, between DNA words. Some people claim that there are hard spacing rules. Some people claim there are no spacing rules. And most of this has been done observationally. You just take, you scan sequences, and you look for enrichments of different patterns in the genome, and you hope for the best. We can use our model as a predictive oracle to actually perform synthetic experiments. So we can, for example, just create synthetic DNA. We can insert 2 DNA words. We can move their positions relative to each other. We can also take real genomic sequences that have these words, and we can mutate them one by one. So this is very analogous to basically biochemistry experiments that are done in the lab and genetics experiments that are done in the lab, right? So we do in-silico versions of these. I'm going to show you 2 of these really quick. Here's an example of 2 DNA words. We just want to understand their spacing syntax. Okay, well, the impact of spacing between these 2 words, how they influence binding of these 2 proteins. So we're showing 2 proteins, and these 2 proteins are known to kind of cooperate in interesting ways. I'm going to see if there's some spacing constraint between these and how they affect binding. So what you're seeing here is an animation, and all we're doing is moving this this DNA word towards this DNA word while keeping the background constant. And we're just using the models to predict binding shadows of these 2 proteins, right? And so what we can do is a function of spacing on the x-axis. We can plot on the y-axis the change in the strength of the shadow, right? That's the y-axis. And we can do this for the 2 proteins independently. So we have 2 different models. And we see something really fascinating. You see the red protein doesn't care. As you change the spacing, its binding, it stays pretty much the same. So it really doesn't care where this gold protein nanog sits relative to it. If you look at the nanog protein, on the other hand, it actually responds pretty heavily. Like there's clearly a cooperative effect. And you can see this enhancement of binding exponentially rising. And then as you get the motifs closer to each other, you see this beautiful periodicity kick in. And that periodicity happens to be exactly 10.5 base pairs, which happens to be the helical turn of DNA. So DNA is a double helix, and one helical turn is 10.5 bases. What this is telling us is that evolution has found a soft syntax. You can see sort of hard syntax. There's not one spacing that works. the model claims that there is a soft syntax that drops exponentially as a function of distance up to 150 base pairs, and there is a preferred spacing. When the two motifs are on the same side of the helix, the two proteins can cooperate. When the two motifs are on the opposite side of the helix, you do not have cooperativity, right? This is really beautiful, like a neural network trained or just on data without knowing any biology has figured out something very fundamental. First of all, asymmetric causal relationship between two proteins mediated through sequence, and a beautiful soft spacing syntax that has learned a helical constraint, right? So it's really cool. Now we can perform the same experiment without using synthetic sequences. We can actually use genomic sequences that contain the same two DNA words. And we can now look at what happens in the genome if we just mutate words and look at effects. So here, these are actual genomic sequences. And here you can see, this is the DNA word for OC4. This is the DNA word for nanog. We can mutate this DNA word, and the model predicts a complete destruction of binding of both proteins. We can mutate the other DNA word, and the model predicts no effect on the red protein and a substantial attenuation of the nanog protein, right? And we can do this for all elements that contain the OC4 and the nanog word, and we can average over those positions. So we can once again plot as a function of distance the effect of spacing syntax on binding, this time in genomic DNA, right? And we again see that same effect replicated, but a little noisier. You can again see the OC4 protein doesn't care, and the nanog protein sees its exponential rise with a sort of a helical periodicity. The reason this is not as clean as what we saw previously is because these sequences are chock full of various DNA words. So even though we are averaging over the genome, we can't marginalize out the effects of just these two words very effectively if you only look at genomic sequences. By using the synthetic sequences, we can really look at just the marginal effects of those two DNA words. Now, of course, that the neural network could be just making stuff up. So luckily, we have experimental tools that allow us to do the same kind of mutations and experiments in the genome itself. So our collaborator, Julia Zeitlinger's lab, she actually took our models, our predictions, and she actually replicated the mutations that we predicted would have specific kinds of effects in the lab through real CRISPR experiments. And so I'm going to show you a few of them. This is one locus. You can see the model predicts this as the binding profile for the SOX2 protein. And this is the actual observed profile. So they look really quite similar. The model predicts this DNA word is very important. And the model says if you break these T's and you make them into A and G, you should see a complete attenuation of binding at this position and an attenuation of binding at this position for the same protein. And we perform the experiment, that's exactly what we see. We see complete destruction right here and substantial attenuation at this position. Now, what we can do is the same, we're going to make the same mutation, but now we're going to measure the impact of the mutation in this SOX2 binding word on another protein, nanog, right? So there's two proteins binding. This happens to be the binding word of SOX2. When we mutate, again, make the same mutation and this time measure or predict nanog binding, you can see a pretty big impact. This shows an indirect effect. SOX2's binding to its DNA word influences nanog's binding, right? Now we can do the reverse effect. That happens to be, hold on one second. Messed up the animations. Oh, I have the wrong slide here, but this golden line right here actually happens to be the nanog word, and we can mutate it the same way. And when we see that, the model predicts that if you mutate the nanog word, it has no effect on SOX2 binding. Again, the asymmetric causality. When we perform the experiment, that's exactly what we see. Unfortunately, I don't have the slide here. I have the same slide repeated twice, so my apologies for that. But effectively, what we're able to do is perform real CRISPR experiments to validate these predictions. Okay. So this is very nice because we can use basically the model as in-silico oracles. We can perform thousands of these syntax experiments, and we can really start tearing apart all the rules of how sequence motifs and spacing constraints, density, combinations, all of this influences binding of thousands of proteins. And these proteins, of course, regulate gene activity, right? Now, this is sort of a way to understand just natural syntax, right? We're not looking at natural genetic variants yet, but we can perform the exact same kind of mutation experiments with real genetic variants. So here's an example of an important genetic variant. Some individuals have a C at this position, and this is previously known to actually affect binding of a particular protein. Okay, so we train a neural network model on that protein. The model has never seen genetic variation; it's just trained on the genome. And then we use a model to predict the effects of the two versions of that variant. This is the C version. It predicts a kind of an interesting profile. When we switch it to the G version, you can see this massive amplification of the binding signal, right? So this is great. It validates, this variant has been known to be affecting binding of this protein and is also associated with autoimmune disease. The model correctly predicts that this variant has a pretty big impact on binding of this protein. But as I mentioned before, we can use the model as a lens and use attribution methods to infer how this variant is affecting binding. And it tells a really interesting story. When the sequence has a C at this position, you can see the DNA word is basically inactive. There is another DNA word right here to the left that drives this weak signal. As soon as you switch the C to a G, it creates this really strong DNA word. And this DNA word and this DNA word, they cooperatively affect binding and they massively amplify binding. So we get a very nice interpretation of what exactly might be happening, and it can allow us to design experiments to test these hypotheses, as I showed you before. So we applied this to various diseases like here's an example of Alzheimer's disease, right? So this is what's called a genome-wide association study. And each point here is a genetic variant. There are millions of these. And the y-axis shows the association of that variant with Alzheimer's. So you can see most of the variants end up doing nothing, but there are these long towers, right? This is called a Manhattan plot because it kind of looks like the skyline of Manhattan. These towers of variants, these are loci that appear to have strong statistical association with Alzheimer's disease, right? So we can go into any of these loci and start dissecting which variants in this loci are actually functional. So I'll show you one such example. Here I'm showing you these kinds of molecular profiles in almost 60 cell types from the human brain. And they look beautiful. They can really capture these elements and their activity. Here you can see an element that is only active in oligodendrocytes. So there's very cell type specific activity. And what we can do is train models on each of these. So we have a model for every cell type. We can take any genetic variant of interest and push it through the model and predict the delta effect of the two versions of any variant, right? And so here's an interesting locus. There are about 165 variants in this locus. The causal variant is hard to pick up just from an association study due to various correlation properties amongst these variants. We can use our models to pinpoint exactly one variant right here amongst these 165. That happens to fall in this very cell type specific element in oligodendrocytes. This element happens to loop and regulate this PICAM gene as shown by this data. And then we can use a model to interpret what the variant is doing. And what the model tells us is this variant is breaking the DNA word of a protein called FOS. So we can produce pretty interpretable predictions of what these variants are doing. We can fine map the causal variant. We can say what it's potentially doing, disrupting the binding of FOS, thereby dysregulating the PICAM gene in a very specific cell type, oligodendrocytes in the brain. And luckily here we had some validation data that supports our hypothesis. We've done the same thing in autism. These are de-novo mutations. These are not germline variants that are transferred across generations. These are de-novo mutations in individuals that have autism spectrum disorder. We can even score those. This time we're using data from fetal brain. Okay, so that the previous data was from adult brains. Again, we can dissect cell types, hundreds of cell types in these fetal brain developmental time points. And then we can use our variants to again score mutations, and we can pinpoint very specific mutations and specific control elements and specific cell types affecting specific genes. We've also done this for congenital heart disease. And just to cut the story short, here we've actually managed to perform CRISPR experiments in stem cell derived cardiac cells. So we have very specific predictions, like this mutation disrupts binding of the ETV protein in a control element next to a specific gene in arterial endothelial cells. So we can make these endothelial cells in the dish, and we can install these mutations, and we can see that the model's predictions end up validating extremely nicely. They are actually affecting expression of the target genes by very, very substantial amounts. All right. So I'll just stop there. And to summarize, I showed you how we can use large-scale molecular profiling datasets to start deciphering genome function. These datasets serve as really nice inputs to train neural networks to map DNA sequence to these molecular profiles. They're able to do these with really amazing accuracy. And we can use simple interpretation methods and use the models as oracles. We can do simulations, all kinds of exciting things to decipher functional DNA letters, words, and syntax. The models can be used to design experiments, as I showed you, actual genome editing experiments. And they're starting to provide interesting clues for therapeutic interventions in rare disease and common disease and so forth. And so with that, I'll just thank my lab. We are a purely computational lab, and we cannot do any of this without our awesome group of collaborators and our funding sources. Thanks.
Thank you very much, Anshul. Is there any quick question?
I guess I have a quick question. Sure. Anshul, you didn't say anything about relative strength of these binding to different sites. Does that matter?
Yeah, so one exciting thing we've recently been able to do is, again, all our models are quantitative. They're predicting these profiles at single base resolution. So we can really quantify effect sizes and so forth and link them back to biophysical concepts like thermodynamic affinity and so forth. And one thing that's been very exciting is even though we're getting feature attributions and all these exciting predictions, it's been a little dissatisfying that we can't put like actual units on these in terms of like biophysics. But luckily, what we've been able to do is we have a paper coming out next week where we show that the neural networks in fact learn delta G's implicitly. And we're able to extract these thermodynamic affinities straight from the neural networks trained on in vivo data in the genome. So that's super exciting because now we make a connection between these completely black box models with classic thermodynamics, which means in the future, we may also be able to parameterize these models more excitingly. Like we can actually have, like, imagine having, neural network units that actually are thermodynamically inspired. They actually like have nice, sigmoid functions that capture, relationship between concentration, affinity, all of these interesting properties. So we will soon be able to start putting very interesting units on these predictions, which actually will resonate very well with the biophysics community.
That's great, Anshul. And I've got more questions about that later for the panel too. Thank you very much. And we'll see you about 4:30 or just after that for the panel. Thank you.